Good morning and welcome to the Capricor Therapeutics DMD Data Conference Call. All participants will be in listen-only mode. Should you need assistance, please signal a conference specialist by pressing the star key followed by 0. After today's presentation, there will be an opportunity to ask questions. To ask a question, you may press star, then 1 on your touch-tone phone. As a reminder, this presentation contains forward-looking statements and information that are based on the beliefs of the management of Capricor Therapeutics, Inc., Capricor, as well as assumptions made by and information currently available to Capricor. Statements in this presentation regarding the efficacy, safety, and intended utilization of Capricor's product candidates, the conduct, size, timing, and results of discovery efforts and clinical trials, scope, duration, validity, and enforceability of intellectual property rights, plans regarding regulatory filings, future research and clinical trials, plans regarding current and future collaborative activities and the ownership of commercial rights, future royalty streams, and any other statements about Capricor's management team's future expectations, beliefs, goals, plans, or prospects constitute forward-looking statements within the meaning of the Private Securities Litigation Reform Act of 1995. Any statements that are not statements of historical fact, including statements containing the words believes, plans, could, anticipates, expects, estimates, should, target, will, would, and similar expressions should also be considered to be forward-looking statements. There are a number of important factors that could cause actual results or events to differ materially from those indicated by such forward-looking statements. More information about these and other risks that may impact our business are set forth in our annual report on Form 10-K for the year ended December 31st, 2013, as filed with the Securities and Exchange Commission on March 31st, 2014, and our Amendment Number 1 to Registration Statement on Form S-1, as filed with the Securities and Exchange Commission on May 23rd, 2014, and in our quarterly report on Form 10-Q for the quarter ended September 30th, 2014, as filed with the Securities and Exchange Commission on November 14th, 2014. All forward-looking statements in this presentation are based on information available to us as of the date hereof, and we assume no obligation to update these forward-looking statements. Please note this event is being recorded. I would now like to turn the conference over to Drs. Linda Marban and Eduardo Marban. Please go ahead.
Good morning. This is Linda Marban, the CEO of Capricor Therapeutics. We are very pleased this morning to welcome you to this call, and it's something we have been greatly anticipating, which is the now release of the Duchenne Muscular Dystrophy preclinical data by Eduardo Marban and his colleagues at Cedars-Sinai Heart Institute. In the next moments, I'm going to turn this call over to Eduardo, who is going to give you a priority and specific look at the data itself. He will walk you through it and explain to you why he is so excited by the findings in Duchenne muscular dystrophy cardiomyopathy. To this end, you will now understand why we are entering a preclinical clinical program for this therapeutic. Thank you very much. And Eduardo, I'll turn it over to you.
Good morning, everybody. The data I'm going to show today were the substance of a presentation that was embargoed until yesterday afternoon at the American Heart Association and contains some proof of concept efficacy and safety data for a mouse model of Duchenne muscular dystrophy and the use therein of a cell type that Capricor is developing, the cardiosphere-derived cell. So the concepts that underlie my talk this morning are the following. First, that cell therapy with CDCs, also known as CAP1002, can regenerate the irreversibly scarred heart. These are published human data from the Caduceus trial. I'll go on to show you, or to mention the fact that safety has been demonstrated with allogeneic CDCs, the CAP1002 product, in the All-Star Phase 1 data. show you the preclinical rationale for the delivery method that we anticipate using as we move forward to clinical studies in the Duchenne muscular dystrophy cardiomyopathy space. And finally, the substance of the talk, which is the evidence for disease-modifying activity in the MDX mouse model of Duchenne muscular dystrophy. So as we reflect on what cardiosphere-derived cells are and what constitutes their unique properties, first of all, they have a distinctive antigenic profile in which all cells are positive for an antigen called CD105, which is a TGF-beta receptor accessory subunit, also known as endoglin. They're all negative for hematogenic markers, including CD45. And the cells have been demonstrated by forensic DNA methods to an accuracy of or resolution of 1 in 40,000 to be of intrinsic cardiac origin, not arising from hematogenous seeding into the heart. They're not mesenchymal stem cells, fibroblasts, myofibroblasts, or cardiomyocytes on the basis of their antigenic appearance and gene expression patterns. They do, however, shrink, scar, and increase the amount of viable myocardium in models of ischemic injury. This has been demonstrated with human CDCs in SCID mice, mouse CDCs in mice, rat CDCs in rats, pig CDCs in pigs, and human CDCs in early phase clinical trials. The direct comparison of the CDC to other clinically applied cell products in a parallel manner with blinded analysis has shown this particular cell type to be functionally superior in terms of the improvement of global left ventricular contraction, as well as in terms of structural benefits and paracrine factor secretion. Finally, the cells are multipotent and clonogenic. Therefore, they fulfill the criteria for cardiac progenitor cells, but the mechanism of action appears not to involve canonical stem cell-based differentiation and implantation, as these are not necessary for long-term benefit. The Caduceus results, in which we use contrast-enhanced MRI to measure living heart muscle and scar, are summarized in the slide that you see right now. What you see right now are the changes in scar mass on the left and the changes in viable mass on the right in each patient added up and presented as pooled data. And in the control subjects, which are the ones shown here, there's basically no deviation from zero in either change in scar mass or in change in viable mass at 6 or at 12 months. These data from the standard of care controls followed longitudinally fulfill the pessimistic dogma that scar mass, once formed, does not go away on its own in a heart attack, and viable mass, once lost, does not regrow spontaneously. We, however, look at the data in the CDC-treated. This is our first in human trials, an autologous product. The scar mass at 6 and 12 months decreased dramatically, and at 12 months the decrease in scar mass amounted to about 12 grams on average. This was in a setting of an infarct or a scar mass that began approximately at 24 grams. So about half of the scar has resolved by 12 months, unlike the controls, which didn't change. Even more notable from a mechanistic point of view is the increase that we saw in viable mass, which amounted to 22 grams at 1 year. The regrowth of living heart muscle in a setting where the loss of living heart muscle is supposed to be permanent after a major heart attack gave proof of concept for the idea that the cells are able to mediate therapeutic regeneration, that is, the purposeful replacement of living heart muscle instead of substituting for dead heart muscle. We were very encouraged by these results from a proof of concept level, but we recognized in the process of executing the trial the limitations of the autologous approach. It works, but it's tedious. The timing constraints are built in because between the harvest and the manufacturing, there's an obligatory 3 to 5 weeks of waiting, so that prompt therapy can never be executed. It's expensive. Each manufacturing run is a one-off. It's risky in the sense that the harvesting procedure itself introduces some risk because of an endomyocardial biopsy. Even greater would be the irony of going through a harvesting procedure that's somewhat risky and then facing the possibility of manufacturing failure, which happened 9% of the time in the Caduceus trial. Not an overwhelmingly high number, but one that we would want to avoid altogether. Because each manufacturing run is racing the clock to produce a viable product, there's not the time to do quality assurance and quality control that one has when one has the luxury of producing a master cell bank, for example. There's inescapable variability in cell potency related to the human being of origin. If the cells came from a 55-year-old man with diabetes, they're going to go back into a 55-year-old man with diabetes, and they may have some of the limitations inherent to that legacy. So we wondered if allogeneic therapy was safe and effective, and in fact we have extensive preclinical data to show that that's the case, and we now have demonstrated safety and preliminary efficacy signals in the All-Star Phase 1 study. I won't belabor that because it's been the subject of previous conversations, but I'm happy to answer any questions that the group may have at the end. Finally, we contemplate a novel delivery method for the widespread delivery of the cells throughout the heart in situations that require complete coverage. In the Caduceus and All-Star trials, the objective was to treat a focal myocardial infarction that affected only one vascular territory in the heart, and therefore we reasoned that it was sufficient to do single vessel delivery of the therapeutic, and in fact that was shown to be effective. In diseases such as heart failure that affect the entire heart, we think it's important to go into all coronary arteries sequentially and deliver the therapeutic so that you get good myocardial coverage. And in fact, we have preclinical data in the porcine model, which has been very successful in terms of convincing the FDA of both efficacy and safety preclinically in 3 INDs to date, that not only do you get pan-myocardial coverage, but also there's no complications in terms of reduction of flow or leakage of biomarkers. Here we compare the safety aspects in terms of reduction of flow and biomarkers with sequential stop flow delivery, in which we actually inflate the balloon in each artery, or in continuous flow, which is our preferred method, in which it's a non-occlusive approach, and the non-occlusive approach had no complications whatsoever. And in data that I'm not showing you, showed a very high efficacy signal. So we've used those porcine data to support the dynamic trial, which is a soon to begin Capricor trial that stands for dynamic here stands for dilated cardiomyopathy intervention with allogeneic myocardial regenerative cells. And in order to get into dynamic, one has to have ischemic or non-ischemic dilated cardiomyopathy with a left ventricular ejection fraction less than 35%, so substantial contractile failure. The therapeutic here will be allogeneic CDCs, CAP1002, with non-occlusive intracoronary infusion in 2 to 3 vessels. We'll infuse into as many vessels as are technically accessible to the operator. It's actually, this is a misprint, it's actually a phase 2A because the therapeutic will have been in humans already because of the All-Star trials, that we're just trying a new patient population. Phase 2A open-label 14 single-center study, and this has been supported by an NIH grant to Capricor. We expect first patient in before the end of this calendar year. So let me show you the substance of yesterday's presentation at the American Heart Association, and this has to do now with the disease-modifying activity in MDX mice. I'm on slide 10 and advancing to slide 11. So slide 11 shows the clinical profile for Duchenne muscular dystrophy. This is an X-linked recessive disorder. So women are carriers, but only male children are affected. The major complications associated with Duchenne muscular dystrophy are related to myopathy, either affecting the skeletal muscle, so skeletal myopathy, or to a cardiac myopathy or cardiomyopathy. The skeletal muscle weakness is apparent from age 3 usually, and there's progressive weakness associated with wheelchair dependency by age 13 often, and then the cardiomyopathy is apparent anywhere from age 6 to age 20 of onset, but usually all of the patients studied who are 18 years or older have manifest scarring within the heart and often a reduction in the global ejection fraction. It starts with a posterobasal fibrosis in the back of the heart and proceeds to global dysfunction and conduction abnormalities, and this is often the cause of death in these young adults or children. There's also a smooth muscle myopathy with vascular dysfunction and urinary and GI symptoms. But from a practical point of view, most patients in the modern age of therapy die either of heart failure or of respiratory insufficiency, and heart failure associated with DMD is a relatively neglected endpoint in clinical trials, and one that we will target here. So to review the pathophysiology of Duchenne muscular dystrophy, it's a monogenic loss of function mutation affecting the dystrophin gene. Dystrophin is a kind of a protein shock absorber that links the cytoskeleton to the surface membrane, and when this is disrupted, the cell membrane becomes fragile. This is not a big deal in a liver cell or a skin cell, but it's a big deal in muscle cells that are constantly contracting, leading to membrane breaks and leakage of extracellular calcium into the cell, among other things. But extracellular calcium normally has a 10,000-fold concentration gradient across the surface membrane. The inside of the cell defends against high intracellular calcium because calcium is an important signaling molecule. When it gets out of control, there's proteolysis, oxidative and nitrosative stress, and inflammation. And so it's the consequences of increased intracellular calcium that appear to then lead to the muscle proteolysis, apoptosis, fibrosis, and involve the heart failure and the skeletal muscle among its targets. So the rationale for using CDCs in Duchenne muscular dystrophy have to do with their signature MOA properties, which include antioxidative action, anti-inflammatory action, anti-apoptotic principles. They also are important in attenuating remodeling after injury, and we found them to be capable of mediating the regeneration of the heart. And when you think of the pathophysiology of Duchenne muscular dystrophy, this cocktail would appear to be well suited to address the central therapeutic lesions. There's intense oxidative and nitrosative stress, inflammation, accelerated programmed cell death or apoptosis, resulting in remodeling of the ventricle, and loss of cardiac myocytes, so the regenerative action would be important there. So the hypothesis is that CDC administration would be beneficial in retarding or reversing DMD, and we tested this in the MDX model, which has been a very robust preclinical model leading directly to a number of clinical studies in the past. The data on slide 14 show 3 indices of cardiac function globally in the heart. In the left-hand panel is shown ejection fraction. And the top line that tracks along at 70 are wild-type animal controls that have been injected just with a placebo in the heart. Their ejection fraction remains about 70% throughout the follow-up of 3 months. And the lower 2 lines are animals with Duchenne muscular dystrophy phenotype. These are MDX mice that are studied at 10 months, at which point they have already developed some loss of cardiac function. You can see that the baseline heart ejection fraction here is 60% instead of 70% in the controls. And then the 2 lines thereafter bifurcate. The bottom line with the asterisk on it is the time course of loss of cardiac function progressively over 3 months of follow-up in MDX mice that were treated only with vehicle. So this shows the natural history of the heart disease in these animals is one of progressive loss of function. Instead, in the animals with the solid squares, those were treated with intramyocardial injection of CDCs at the day right after baseline. And those animals showed an improvement, a net improvement in cardiac function, which was sustained relative to control for the entire 3 months of follow-up. This consisted in the middle panel of both an effect on end diastolic volume as well as a marked decrease, more pronounced in end systolic volume in the right-hand panel. So that shows that basically the squeezing power of the left ventricle is increased. We find when we look at the hearts histologically that control hearts stain brick red with this Mason trichrome stain. That means they have no scar. MDX hearts treated with vehicle, looked at here 3 weeks after injection, have a substantial amount of fibrosis as seen by the areas of blue and gray within the otherwise red heart. But the amount of blue and gray markedly decreases. It essentially goes away in animals that have been treated with CDCs. And independent confirmation of the antifibrotic effect is to be seen in panel B in slide 15, in which case we've actually used immunoblots to measure the immunoreactive collagen 1 on the left and collagen 3 on the right. And you can see that this protein, which is the principal component of scar within the heart, is markedly increased in the MDX mice that are treated only with vehicle, but intensely inhibited in the CDC-treated animals. So there's less fibrosis both by pathology and histology as well as by measurement of protein density. When we see whether there's any functional benefit globally within the animals, we find something interesting. And this is a treadmill assay in which we exercise the mice to tolerance. At some point, the mice give up. They're just too tired and tolerate an electric shock at the end of the treadmill. The top line, orange-colored, is the ability of a control wild-type mouse to exercise. These mice will go for 700 meters before giving up. On the bottom line is the result in an MDX vehicle-treated mouse population. They can only go about 250 meters before they poop out. But note that the middle line in blue with the asterisks shows that function, the ability to walk, goes up. It almost doubles in animals that have been treated with the therapeutic, and that effect is sustained for at least 6 weeks after therapy. This means that the heart treatment was enough to basically double the ambulatory capacity of these animals. If we look mechanistically at what's going on, some of the targets that we have explored include the transcription factor NRF2 that is a central controller of antioxidant pathways. We expect NRF2 and its downstream effectors to be increased in any situation in which the heart experiences oxidative stress. And in fact, when there is excessive oxidative stress, increase in NRF2 is a valid therapeutic principle. We find, this is now slide 18, if we look at the images on top, which show immunolocalization of NRF2 as red dots, we find the control hearts have some NRF2. It's mostly in the cytoplasm. The MDX-treated hearts show more localization of the NRF2 towards the nucleus where it's active, but the CDC-treated MDX mice show a tremendous amount of localization of NRF2 to the nuclei. And these conjectures are borne out by further analysis at the immunoblot level. On the lower left-hand panel, you see the phosphorylated form, which is the active form of NRF2. And on the right lower panel, you see the localization into the nucleus. And both of these are modestly increased just in the disease as a compensatory process, the middle panels. But you can see that the right-hand bars labeled MDX plus CDC show marked upregulation both of the cytoplasmic and the nuclear translocated forms of NRF2. So this is a compensatory response that is markedly upregulated by treatment with CDCs in a salutary manner. So in the next slide, which is slide 19, we see that CDC treatment heightens the activity not only of NRF2, I've shown you that, but of important downstream targets. The upper left-hand panel shows hemoxinase 1, which is one of the antioxidant targets downstream of NRF1. That's increased. And then there's the lower left panel shows superoxide dismutase 1, which is a very well-known antioxidant enzyme, again markedly upregulated by treatment with CDCs, as is catalase, another antioxidant enzyme, and then further confirmed by the SOD2 isoform in the lower right panel. We find also very interestingly that NRF2, through coupling downstream to mitochondrial transcription factor A, improves some of the central lesions in mitochondrial bioenergetics and abundance that are part and parcel of the central lesion of the muscular dystrophy process. Here on slide 20, we see in the first immunoblot on the left, again, the nuclear NRF1, and then we see in the next panel that's labeled nuclear MTTFA, an enormous upregulation in mitochondrial transcription factor A, which would favor mitochondrial biogenesis and regeneration of these organelles, which are decreased in muscular dystrophy. You can also see that the cytoplasmic form is upregulated, and on the right-hand panel in that middle series, mitochondrial DNA copy number per cell, you can see that's markedly decreased in vehicle-treated MDX mice, which tells you that the number of mitochondria is markedly attenuated as part of the disease process. But this number is brought back almost towards normal levels by CDC treatment. The bottom left immunoblot shows on the left the control abundance of a number of proteins that are involved in the mitochondrial respiratory complex that actually do the work of the mitochondria to synthesize ATP. And you can see in the middle 3 lanes, which are from MDX vehicle-treated animals, a marked downregulation of the abundance of these mitochondrial respiratory complex proteins, which comes back very much towards normal after CDC treatment. So CDCs appear to restore mitochondrial copy number, as well as mitochondrial respiratory complex proteins as a result of upregulation of mitochondrial transcription factor A. We also see at an ultrastructural level that the abnormalities associated with mitochondria in the MDX mice, which include excessive mitochondrial length, bottom left, rounding of the cristae within the mitochondria in the bottom middle, or the number of these cristae stacks per mitochondria, which is decreased in the MDX mouse. All of these abnormalities are corrected by treatment with CDCs. And we've recently discovered as well that the respiratory abnormalities in the mitochondria are shown here as oxygen consumption rate in isolated mitochondria. The yellow line is in MDX-treated mitochondria, MDX control mitochondria that has been treated only with vehicle. And the top two lines, which are indistinguishable from each other, show control mitochondria and MDX CDC-treated mitochondria. So we've succeeded on slide 22 here, we've succeeded in correcting a central lesion of mitochondrial respiration by CDC treatment in these animals. Finally, the amount of cardiomyogenesis, that is the amount of new heart muscle that's generated, is shown here in slide 23. The top two panels indicate the amount of mitosis that's going on, increasing in Ki67, an increase in ORRB, which is a marker of cytokinesis. Each of these two are modestly increased just by the disease itself as a compensatory mechanism, but very much upregulated in an MDX plus CDC-treated mouse model. And you can see in the bottom set of panels labeled C that the number of endogenous stem cells that are tracked to the myocardium, here indicated by a combination of the c-KIT antigen and the transcription factor, the cardiac-specific transcription factor NKX2.5, markedly increased by CDC treatment. So in slide 23, there's three pieces of evidence that cardiomyogenesis, or the origin of new heart muscle, is increased. On the top is an increase in mitotic markers, in the middle panel, an indicator of active cytokinesis, or ORB, and in the bottom, tracking of endogenous cardiomyocyte precursors to the site of injury. So moving on to slide 24, this summarizes the observations in terms of therapeutic mechanisms of action or therapeutic observations. Injection of CDCs into the MDX hearts improves the global function of the heart, decreases fibrosis, improves exercise capacity, exerts very potent antioxidant effects. It reverses the central abnormalities in mitochondrial abundance, structure, and function, and increases cardiomyocyte proliferation while also increasing activation and recruitment of endogenous repair. So these data have motivated the trial acronym that is in slide 25, which we hope to pursue in the near future, and that's the HALT cardiomyopathy progression in Duchenne trial, or the HOPE Duchenne trial, which Capricor is actively planning. We had a KOL meeting a week ago to initiate the clinical protocol planning process. The CMC sections for an IND will have already been completely prepared because of the All-Star experience, and we have a very robust preclinical package ready. So that concludes my formal presentation. I'll turn it over for further moderation to others in the Capricor team.
So thank you, Eduardo. That was very, very informative and helpful. For those of you that didn't follow completely the science, I think the most notable effect are the molecular changes in mitochondria, which leads to the potential not only to improve cardiac muscle but also skeletal muscle, as Eduardo said. I think we're going to now turn it over for questions. Eduardo stands ready to answer any of your questions, and so we'll turn it back to the call center.
Thank you. We will now begin the question and answer session. To ask a question, you may press star then 1 on your telephone keypad. If you are using a speakerphone, please pick up your handset before pressing the keys. If your question has been addressed and you would like to withdraw from the queue, you may press star then 2. At this time, we will pause momentarily to assemble our roster. The first question will come from Kevin DeGeeter of Ladenburg. Please go ahead.
Good morning. Thank you, Linda. Thank you, Eduardo, for a very informative call. A couple of quick questions. I basically want to get a better feel for how one should think about potential subpopulations and patient response as we move into clinical development. I guess my first question is just to better understand. You identified a number of potential biomarkers, molecular biomarkers, in the presentation. Which of those do you think might be most informative as we move into clinical development? And do you think there's a role here for perfusion and other perhaps imaging-based biomarker analysis as well on the clinical development program?
Linda, would you like to answer that? I'm certainly happy to do so as well.
Yeah, go ahead, Eduardo. I think you're the appropriate one for that question.
So what we know about biomarkers in Duchenne cardiomyopathy is as follows: that the children have normal heart function until they develop some level of fibrosis in the heart. And once they develop that level of fibrosis, the heart function begins to decline progressively, ultimately leading to death. That transition from no fibrosis to fibrosis can occur as early as 6 or 7 years of age or as late as 18 to 20 years of age. And that's been identified, that sequence has been identified by sequential magnetic resonance imaging in patients with Duchenne muscular dystrophy. So a lot of the major centers that follow substantial numbers of these patients now begin doing annual or somewhat frequent MRIs with contrast enhancements on IV to make the scar light up in the heart. And once that happens, they know that there's some indication of a prognostic importance, because once the fibrosis begins, the functional deterioration is progressive and inexorable. So we envision using late gadolinium enhancement as an indication of fibrosis, both as an inclusion criterion for the trial, as well as a measure of efficacy in the trial. We will simultaneously be measuring a number of serum biomarkers, including high sensitivity troponin I, creatine kinase, and others, to see if there are also further measures of efficacy. But the major phenotyping measures will be related to fibrosis as measured by contrast-enhanced MRI, clinically indicated echocardiography, as well as serum biomarkers.
Great. Maybe one more for me, then I'll get back in the queue. Can you just clarify whether or not we'll be looking at capturing patients that also have dilation, and just whether or not the data from the MDX mice gave us any insight as to whether cardiomyopathy, the dilation had any impact on responsiveness to the CDC cells?
Fibrosis seems to be fibrosis and decline in systolic function, the ability of the heart to actually contract rather than dilatation, appear to be the earlier hallmarks of Duchenne cardiomyopathy, and the dilatation happens more in the end-stage disease. Even though we will certainly be looking for dilatation or an increase in end diastolic volume as one of our response measures, We believe that there's more to be learned in terms of signals of efficacy looking at fibrosis and end systolic volume, and ejection fraction. I would just say parenthetically, I didn't say this before, but this is the only MDX preclinical study of which I'm aware in which we actually have seen a net improvement in cardiac function. The best, or fibrosis or any of the other endpoints that we've measured, the best that's been achieved previously is some attenuation of progression. So I think there's room for hope, at least, that there might be some actual improvement in some of these parameters. Obviously, we won't know until we test it, but that would be our hope.
Great. Thanks so much.
Thanks, Kevin.
The next question will come from Jason Colbert of Maxim Group. Please go ahead.
Hi, this is actually Jason McCarthy for Jason Colbert, and I have more of a molecular question. And my question is, if CDCs are upregulating NRF2 and they're reducing inflammation, and they're improving mitochondrial function through mitochondrial transcription factor A, is there also an upregulation of SDF1 that could, further aid in cardiac repair, possibly by recruiting more cardiac stem cells to the heart or to the damaged tissue?
Right. We have definitely documented that the CDCs increase SDF1 levels in target tissue. We haven't looked at this specifically yet in the MDX mouse, but you correctly identify that as one of the MOAs that's operative here.
Okay, and also, have you identified any other master regulators of transcription that have been up or down regulated, maybe possibly related to angiogenesis or other cardiomyogenesis?
We have been looking primarily at the pathways that I've shown you, but I can also mention that nitrosative stress pathways are attenuated, as are, and that among the protein factors that seem to be increased as a consequence of CDC therapy, VEGF is locally increased in the heart. That's about as far as we've gone so yet.
Great, thank you.
The next question will come from Ted Tenthoff of Piper Jaffray. Please go ahead.
Great. Thank you. Good morning. Can you hear me okay?
Yeah.
Yeah, we can hear fine.
Excellent. Great. So first, I just really have to applaud your efforts to focus on cardiac disease for Duchenne boys, because with all the effort in the industry focused on skeletal muscle, we're still not sure that we're actually going to have survival benefits. So I really appreciate you guys tackling kind of the core underlying problem here. And Dr. Marbán, I really appreciate you going into all that preclinical data. I guess my first question had to deal with sort of kind of a very high-level basic question, and it has to do with sort of whether or not these cells are actually engrafting, or whether it is more through a transient process where they are simply excreting factors that are causing all of these beneficial changes.
So, Ted, that's a good question. In data that we showed yesterday at the AHA, but I did not include in today's synopsis, we have shown that the effects of CDCs are mimicked well by exosomes that are produced by the CDCs and can be purified from conditioned media. So it's a cell-free nanoparticle, membrane nanoparticle. And those cell-free membrane nanoparticles contain a distinctive profile of non-coding RNA. Our working hypothesis for the mechanism of action of the CDCs, in fact, in both ischemic models and in DMD, is that the CDCs engraft in the tissue produce exosomes, because you can blunt the therapeutic effects of CDCs if you block exosomal production. And you can mimic the effects of CDCs with exosomes. So we are just beginning to understand MOA as being due to secretion of exosomes that contain non-coding RNAs that then reach target tissues and induce epigenetic reprogramming there that then has long-lasting functional benefits. So that's our current understanding, and indeed it's all indirect. The cells just need to be there long enough to secrete the exosomes and trigger the long-lasting changes in reprogramming.
Well, that is really interesting. So it's a product of the CDCs. And then I guess a follow-up, if I may, in terms of access or perfusion through the heart, and I guess perfusion is not the appropriate term, but you mentioned that with an acute MI, it really is a precise point where you need intervention. With heart failure and or cardiac distress caused by Duchenne, it really is throughout the cardiomyopathy. So how far into the heart muscle are these cells actually getting? And is this a phenomena kind of like the skeletal muscle dystrophin production that we've seen with other DMD therapies, where a certain threshold needs to be achieved to actually maintain cardiac function? For example, we're around a 10 or 15% number in terms of dystrophin production, actually producing enough viable skeletal muscle. Is it something like that, where it's actually a small threshold that maybe needs to be achieved in order to have more profound cardiac benefits?
So to address your first question first, the cells, when they're delivered into the coronaries, appear to lodge at a capillary or precapillary level throughout the heart, and we can measure using a reporter gene like luciferase and grinding up different pieces of heart muscle throughout the heart, that when we do this triple vessel delivery, we get very extensive myocardial coverage. There's no part of the left ventricle that seems to be spared. The cells, at a microscopic level, the cells appear to lodge, fairly distally in the small vessels where they're very likely to be able to secrete their factors into the surrounding heart muscle. The second question had to do with, can you just repeat that? I'm sorry.
Yeah, it's kind of more of a high-level question, and this may be something that we'll just have to see as we get and learn more in human and in the clinic. But when it comes to dystrophin production, I think it's a different mechanism in skeletal muscle. It seems that there's a threshold that needs to be achieved, which is actually not a huge threshold to have a more profound therapeutic benefit. Is there the potential that we may see something similar here with the cardiac muscle tissue as well?
Right. Now, so it's a good question. So we have not done particularly extensive dosing studies in the MDX model per se, but when we do very, very meticulous dosing studies in a pig model, we find evidence that there is some threshold dose that produces, in the case of the pig coronary being somewhere between 6 and 10 million cells, that produces an almost quantal effect. So if you go above that, everything's better, and if you go below that, there's very little efficacy. It's not like a receptor-mediated process, so that's very highly graded. So we believe that there's some level of cell engraftment that leads to sufficient production of exosomes that then target the tissue and to some extent actually lead to a self-perpetuating healing process. And so that, I think what you were intuiting in the question probably is right. We don't believe that the MOA here has anything to do with restoration of dystrophin in the sense that we're looking at that now, but I believe we're generating new cardiomyocytes that are genetically abnormal because they still arise from the host, but they're protected from immediate injury by virtue of being young. It takes a while for them to get sick to begin with, and also by attenuation of the oxidative and nitrositive stress processes.
Great. That's incredibly helpful. Thank you for that update.
Our next question will come from Ren Benjamin of H.C. Wainwright. Please go ahead.
Hi, good morning, guys, and thanks for taking the questions, and congratulations on the progress. I guess just a couple of questions. When I look back at the mouse data, in particular ejection fraction, I noticed that it's a one-time treatment. You appear to see this benefit at week 3, it looks like, and then you get this trend of kind of resuming pretty much like the MDX mouse itself. And so it's kind of a twofold question. One is, it seems like this would be amenable to multiple dosing, and can you do that given that it's intracoronary injections? And when you go into the clinic, how do you guys plan, to be addressing the dosing question?
Right. So we have extensive dosing information with the therapeutic already from the All-Star phase 1 trial. That was single vessel delivery, but we found in the porcine model anyway that we can use the same dose safely in each of the vessels. So we'll basically be doing triple dosing relative to the whole heart, relative to what we've seen in All-Star, and the rationale being that in All-Star there's focal injury caused by a heart attack, and here it's a global process. So our target dose in All-Star and the one that's being used in the phase 2 randomized placebo-controlled trial that's going on right now is 25 million cells. Here it'll be 75 million cells.
Potentially. We haven't really completely decided on a dose, right? We're going to do a dose.
No. So our thinking is that most of these boys will be adult sized or young adults, and that that'll most likely be our target dose. But we will probably or we may include in a clinical protocol some kind of dose escalation. But that in sort of a rough measure is an indicator where we have our dosage target at least. The question of repeat dosing is one that's extremely interesting to us. We're beginning preclinical experiments right now with repeat dosing in the MDX mouse model, and we anticipate that there's nothing durably magical about this therapy. At some point, maybe 3 months, as we've seen in the MDX model, or maybe longer. But we'll be looking for improvement and then subsequent deterioration, and that would be a time when we would contemplate repeat dosing. Most of these individuals will be on steroids, if not all of them, and steroids will give some added measure of protection in terms of any possible immune reaction that might arise to the first dose. But this is obviously the kind of thing we'll be looking for in great detail in this population. Is there the development of any subclinical antibody response and things like that? So the short answer to a very sophisticated question is we will definitely be looking at repeat dosing very carefully and looking for clinical indicators as to when repeat dosing should occur, and we'll try to build that into our initial protocol.
Okay. And then just related to that, and I'm going to the Cardiosphere project that's in preclinical development. Why not, instead of direct injection into the heart, have you put any thought into or thought about using the Cardiospheres and a systemic IV administration so that the potential benefit of these Cardiospheres could be affecting not just cardiovascular muscle, but also skeletal muscle?
Are you thinking, you said Cardiosphere, but are you thinking exosome?
Yeah, I'm sorry, exosomes.
This is a next generation product that we're developing. Linda, would you like to mention something about that?
Yeah, so Ren, also follow up to what Eduardo said before, is, one of the reasons we're going to be using the cells in this first iteration trial will be because we know a lot about the cells. We know how to dose them, we know how to deliver them, and we'll also then be able to gauge when their effect in humans might begin to wear off. And then we'll have the option to either enter repeat dosing with the cells if we feel that that's the appropriate thing for the patient, or at that point we may decide to use the exosomes for repeat dosing paradigms, in sort of whatever reasonable time frame that would allow. We are considering all options, including potentially infusing the cells in such a way to help improve the skeletal muscle. The skeletal muscle observations are hot off the press and very new, and so we have to do some more confirmatory work to determine if that's going to be a viable strategy, but certainly one that has us very excited.
Got it. And just one final question for me in terms of potential collaborations or partnerships. I think it was Ted who mentioned that clearly there are a lot of other players in the space focused more on the skeletal muscle issues and problems. This therapy seems just positioned correctly to be used in combination with either the exon skipping technologies or the null mutation skipping technologies. Have you guys evaluated the potential for a combination of these two types of therapeutics in the MDX mouse model? Do you plan on doing it? And I guess probably more importantly is if you see this as a future, do you think a collaboration or a partnership with some of the existing players is plausible?
Yeah, so I think I'll take that one first, Eduardo, if you don't mind. Yes, one of the things that's got us really excited, Ren, is the idea that this therapeutic can be used probably in parallel to all of the other strategies that are addressing the skeletal muscle myopathies. We don't plan on doing any animal studies ourselves to confirm that, because there'd be no reason to believe that those are not possible, but if the opportunity presented itself, we certainly would explore it at the animal level. In terms of collaborations and partnerships, I think the answer there is that absolutely and always we're considering those opportunities right now. We're really excited with what we're doing. We have a lot of work to do in-house, and so we're focusing hard on our own efforts, but certainly if the right opportunity presented itself at some point, we would definitely entertain that.
The only thing that I would supplement what Linda said is that in all of the exon skipping technologies and nucleotide-based technologies of which we're aware, there's never been any increase in dystrophin in the heart in MDX mice. Yeah. So none of those therapies appear to touch the cardiomyopathy.
Right. Then, and the penetration has always been an issue there, which is why right off the bat I would think a combination strategy would be a great way to move forward. But thank you guys very much. And good luck.
Thank you. Thank you, Eduardo, by the way. That was good.